Welcome back, students. In the previous class, we had studied about the chromosomal theory of inheritance, and we also studied about other various aspects related to linkage and recombination, and we also discussed about the mechanism of sex determination in humans, in Drosophila, in birds, and also in grasshoppers. Now, when we talk about chromosomal theory of inheritance, there we studied that the different traits or characters are transmitted from parents to offspring because of the inheritance of certain chromosomes which carry certain specific genes which are responsible for these characters. So in other words, the inheritance of the chromosome is also causing the inheritance of the genes, and therefore certain characters are transmitted. Similarly, when we talk about certain disorders, there are certain genetic disorders in human beings which are transmitted from parents to offspring. So these disorders are actually, we don't call it as diseases because the pathogens are not involved, but because of certain abnormalities, certain mutations that take place in the genes, so these disorders are caused. So these disorders cannot be avoided in human beings, but we can study the pattern of inheritance of these disorders. Why? Because in human beings there is no controlled crosses. The controlled crosses are not possible in human beings. If we again go back to Mendel's crosses, say for example, at his will, Mendel could cross tall plants with short plants. Similarly, if he wanted to cross plants with yellow seeds and plants having green seeds, he was free to do so. So this is called controlled crosses. The one who is doing experiments can choose the type of the parent. So that is called controlled crosses. But in human beings, we cannot perform controlled crosses. So therefore, we cannot avoid the transmission of these genetic disorders from parents to offsprings. It cannot be avoided. Rather, we can go for analysis of these traits. We can also go for study of the pattern of inheritance of these traits over several generations in a given family tree. So this study of the inheritance of certain disorders in a given family tree, so this is known as pedigree analysis. So in pedigree analysis, basically what we can do, we can study the pattern of inheritance of these characters, whether it is being inherited from father to son or mother to daughter, or whether from mother to both sons and daughters, fathers to sons and daughters, and so on, or whether it skips certain generations, whether it is seen in one generation, in the grandparents' generation, whether it avoids the next generation, whether it is coming up in the grandchildren generation. So in that way, we can study or analyze the pattern of inheritance of these traits or characters, or it could also be called as genetic disorders. So this pedigree analysis is a very strong tool. It is utilized to trace this inheritance of a specific trait, abnormality or disease along number of generations. In order to study the pattern of inheritance of these characters from one generation to the other, we need to first make a family tree, and then we need to see the pattern of inheritance of these characters. So in order to make this family tree, certain symbols we need to understand first. Let us go to those symbols. So these are the various symbols that is used in construction of a family tree. So this symbol represents male, the circle represents female, this one represents sex unspecified, and here you can see square, circles and sex unspecified. All these three are shaded. This means these are the individuals which are affected. That means these are the individuals which have certain genetic disorders or traits or characters. And here we see this is showing mating between close relatives. So this is male, this one is female. So two lines means mating between close relatives if there is only one line, so this means this is marriage, normal marriage, because there is one line, and this here you see there are two lines, means this is meeting between close relatives. That means within a family, if someone marries with each other, so then we have to put this double line. And the next pedigree that we see here, so this is marriage, this is father, this is mother, and then we can see in the next generation there are three offsprings. The first offspring is a male, so we see this is male. The second and third offsprings are females, and all these offsprings they do not carry any genetic disorder. Why? Because it is not seeded. So before we move on into more details of the pedigree analysis, let us also discuss about the genetic disorders. Basically, the genetic disorders are not really diseases, but these disorders are because of the mutation in a certain gene, or the presence or the absence of certain chromosomes. So therefore we call it as disorders. So actually there is no pathogen involved. Pathogen is an organism that causes a disease. So here in these disorders there are no pathogens. It is because of the genetic reasons that these disorders arise. So basically we can divide this genetic disorders into two types. The first one is known as Mendelian disorders, and the second one is known as chromosomal disorders. So this Mendelian disorders, the first type, so this arises due to alteration or mutation of a single gene. So there is mutation taking place in a single gene. So if because of this gene mutation will arise this Mendelian disorder. And why it is known as Mendelian disorder? Because it follows the Mendelian pattern of inheritance. So one such example of this Mendelian disorder is myotonic dystrophy. Again, this Mendelian disorders can be divided into being of four types. One is autosomal. Either the Mendelian disorders are autosomal or sex-linked. So basically, or either it is dominant or recessive. So we can divide it into being of four types. One is autosomal. That means this disorder is being carried, or this mutation has taken place in the autosomes, in any of the autosomes. Okay, because there are two types of chromosome. One is known as autosomes, the other one is known as sex chromosome. When we say sex chromosome, it is X chromosome and Y chromosome. So the disorders can be categorized as being autosomal and sex-linked. At the same time, it can also be categorized as dominant and recessive. So basically here we can see I put it as autosomal dominant, autosomal recessive, then there is sex-linked. That means this is related to either X chromosome or Y chromosome, sex-linked, but at the same time dominant, sex-linked and recessive. So we are going to see whether it is how it is, we can say certain disorder is autosomal or it is sex-linked, or on what basis we can say certain disorder is dominant or whether it is recessive. So we'll come to that later on. So I've also given certain examples of autosomal dominant, which I already referred to. Is one such example is myotonic dystrophy. The autosomal recessive trait, sickle cell anemia, of which we are going to study in more detail after some time. Sex-linked dominant. It is related to sex chromosome, but it is dominant. So example, vitamin D resistant rickets. And sex-linked and recessive character. So example, hemophilia. Now let us come into more details of autosomal dominant trait, that is we discussed myotonic dystrophy. This myotonic dystrophy is characterized by the wasting of the muscles. The person suffers from cataract, endocrine changes that take place in the person. That means these are hormonal changes. Weakness in the lower legs, in hands, in the neck, in the face. So these all are the symptoms of myotonic dystrophy. Now if you see this pedigree, that which I have constructed here, so this pedigree is showing the pattern of inheritance in case of myotonic dystrophy. So this pedigree is of the type which is called autosomal dominant. So it is carried by the autosomes. Okay, we have 22 pairs of autosomes, and 22 pairs means 22 into 2, that is 44 autosomes, and the trait is dominant, means we are going to discuss about why it is dominant. So if you look at this pedigree, then you'll see certain pattern of inheritance. Now some more details of why it is called autosomal. So this order, if you see the pedigree chart which I have shown here, this order is passing from passing from mother to both son as well as daughter, both male as well as female. So it is not located in the sex chromosome, so it is located in the autosomes. So if the character is carried in X chromosome, then it could be X or Y chromosome, then it could be sex linked, but here it is not carried by the sex chromosomes, so therefore it is autosomal. And at the same time here, how do we know? Because the trait is being transmitted to both sons as well as daughters. Again, this trait is called dominant. So why dominant? Because if you see the pedigree, this trait is seen in all generations of a family. So this is generation 1, this is generation 2, it is also there, 2 individuals are suffering, and this is generation 3, so 1 individual suffering here. So the trait is seen in all the generations of a given family, and in other words we can say it is not skipping generations. The trait does not skip generations, so therefore this trait is dominant. Now let us see the other trait, that is autosomal and recessive. So examples of autosomal and recessive trait, as I've already said, is sickle cell anemia. In sickle cell anemia, the shape of the RBC changes from normal, that is biconcave disc shaped structure. Okay, I've shown 2 angles. So biconcave disc shaped structure to sickle celled. So it becomes like a sickle, and because it has changed its shape into sickle shaped, the oxygen carrying capacity of this red blood corpuscles will decrease, and that leads to anemia. So this is a pedigree chart that I have shown for the inheritance pattern of sickle cell anemia. We see these 2 parents, they are not suffering from any disorder and sickle cell anemia. Then in the generation 2, this is generation 1, this is generation 2, G2, we find 2 individuals, 1 son as well as 1 daughter is suffering. Then in the next generation, the 3rd generation, we find 1 individual, that is the first male that is born, is suffering. Then we will see whether this is autosomal or dominant first, then we'll come to whether it is autosomal or sex linked, or whether it is dominant or recessive. So this character, as we know, is autosomal. Why? Because it is being transmitted both to sons as well as daughters. So therefore we should keep in mind that this is an autosomal trait. So the other thing is that this disorder is recessive. Why? This disorder is skipping generation. It is skipping G1, that is in the grandparent generation here. It is skipping. Then in G2 it appears. In G3 also it is appearing. Maybe in the next generation it will skip. So therefore it is not dominant, but it is recessive. So therefore sickle cell anemia is a autosomal, at the same time recessive trait. Now let us see the pattern of inheritance of sickle cell anemia. This sickle cell anemia is controlled by 2 alleles. One is HbA and the other one is HbS. S for sickle cell anemia. And accordingly, because there are 3 types, there can be 3 types of individuals. So the first individual having HbA and HbA, both HbA, so this individual is a normal individual who does not suffer from sickle cell anemia. The second type is a combination of one allele is HbA, the other allele is HbS. So this is why? Because there is one normal allele, so this individual is unaffected. But at the same time, because it contains one allele HbS, it is a carrier. Although it is not affected by the disorder, it can transmit, this individual can transmit the trait to next generation. So therefore we call it as unaffected carrier. The third one, HBS. So because both the alleles are HBS, so therefore the individual is affected, or in other words will suffer from sickle cell anemia. Now I had mentioned all these traits to be Mendelian disorders. So now I'll be showing you how what is the pattern of inheritance of sickle cell anemia. It follows Mendelian pattern of inheritance. So let us see the pattern of inheritance here. So this individual, the female parent having HBA as the alleles, when it is crossed with male parent having HBS, that means this is a unaffected carrier individual. So then the gametes which are formed here, because both the alleles are HBA, so there is only one possibility, one type, that is HBA, and the other, from the other parent, that is the male parent, we see there are two types of gametes, one having HBA, the other having HBS. And now when we put this gametes in the Punnett square, we find combination here. So from females there is only one type of gamete that can be formed. From males there are two types of gametes that can be formed. So this is the genotypic combination. So HBA becomes a normal individual, HBS is a carrier. So we can understand the inheritance pattern of this sickle cell anemia using a Mendelian cross. So let us see what causes sickle cell anemia. So as we have already discussed, so this is a genetic disorder. So this is the biconcave shape of RBCs. So this is a normal hemoglobin gene. So here normal hemoglobin gene with this is the DNA of in the gene, and from this DNA is formed this messenger RNA, the details of which how it is formed we will be studying in the chapter on the DNA, the next chapter. And then this messenger RNA will result in the formation of GLU here. This is an amino acid. So this is a this is a polypeptide chain. These all are different sequences of amino acids which form a single protein or polypeptide. So here this messenger RNA is resulting in the formation of GLU, refers to glutamic acid. This glutamic acid resides in the 6th position of the beta chain of hemoglobin. Now if in the same position here, this T for thymine, A for adenine, ATGC, adenine, thymine, guanine and cytosine. These are the 4 nitrogenous bases that is there in DNA. So this TA at this location, so there is inversion. This nitrogenous base is flipping here, and it forms another combination. So if there is a slight mutation that has, we can say there is a mutation that has taken place. This mutation is known as point mutation. So instead of AT, it has become instead of TA in the previous case TA, it has become AT. So the combination here initially it was CTC, now it is CAC. So therefore the messenger RNA that is formed is different here. So in the first case it was GAG, second case it is GUG. So because of this messenger RNA, then the amino acid that is brought in at 6th position of this beta globulin chain of hemoglobin. So the amino acid brought is valine, VALINE. So this is an amino acid. So here we see instead of the normal, that is glutamic acid that should have been there, because of this mutation it is resulting in the change in the amino acid in the 6th position of beta globulin chain, and because valine is there instead of glutamic acid, now the shape of the RBC will change, and the shape of the RBC will now result in the less oxygen carrying capacity of this RBC, so therefore leading to anemia. Now coming back to this chart, the third category, that is sex-linked dominant. Examples: vitamin D resistant rickets. We don't need to go into details of this in the syllabus. The fourth category, that is very important, that is sex-linked recessive disorder, example of which is hemophilia.